Assalamu alaikum class. Let's start with the muscle physiology. Before starting our lesson, I will give you the learning objectives of our lecture. Uh, by the end of the lesson, you should be able to name the contractile proteins of muscles, and you will be required to enumerate the events which occur during the neuromuscular transmission. Then I will also tell you the role of calcium in the muscle contraction, which is given a special name, and that is called excitation contraction coupling. And you will come to know about certain interesting theories such as walk along theory of the skeletal muscles. And you should also know that what is the mechanism of skeletal muscle contraction. Then I will tell you the differences between different types of muscular tissue. And in the end, we will look at some conditions like rigor mortis and myasthenia gravis. So let's start with this lesson. As you can see, that there are three types of muscular tissue: skeletal, cardiac, and smooth muscle tissue. And I am just assuming that you already know the general functions of muscular tissue, which are producing body movements, stabilizing body positions, storing and moving substances within the body, and also generating heat, which is called thermogenesis. Let's now have a brief look at the physiologic organization of skeletal muscles. The skeletal muscles are composed of numerous muscle fibers. The muscle cell is called the muscle fiber. Diameter of a muscle fiber is around 10 to 80 micrometers. The cell membrane is called the sarcolemma, and it also contains thin collagen fibrils. Connective tissue coat, which surrounds the group of muscle fibers, it separates them into fascicles. You must also understand the composition of the muscle fiber. Here you can see that each muscle fiber is made up of successively smaller subunits, which are called myofibrils. Many myofibrils of each muscle fiber, they are suspended side by side in the muscle fiber, and the spaces between the myofibrils, they are filled with intracellular fluid, which is called sarcoplasm. In the sarcoplasm surrounding the myofibrils of each muscle fiber is the muscle cell endoplasmic reticulum, which is called sarcoplasmic reticulum. Each myofibril is composed of 1500 myosin filaments and double the amount of actin filaments, like 3000 actin filaments for 1500 myosin filaments. The myosin and actin filaments, they interdigitate, and thus they cause the myofibrils to have alternate light and dark bands, which are labeled I and A bands. The light bands, they contain only actin filaments and they are called I bands. The dark bands, they contain myosin filaments and the ends of actin filaments, and they are called A bands. The ends of the actin filaments are attached to Z discs. This Z disc or Z disc attach the myofibrils to one another all the way across the muscle fiber. Skeletal muscle is also called striated muscle because the entire muscle fiber has light and dark bands, as do the individual myofibrils. These bands give skeletal and cardiac muscle their striated appearance. Sarcomere is a crucial concept. Actually, the portion of the myofibril that lies between two successive Z discs, that is called a sarcomere. Wondering what is titin? Titin is a springy filamentous protein which keeps the myosin and actin filaments in place. And it is one of the largest protein molecules in body, extending from Z disc to M line. Titin acts as spring while the length of the sarcomere is changed. And it also acts as a template for contractile filaments formation. Let's look at the myosin filament, or you call it thick filament. Myosin filaments are composed of multiple myosin molecules. The myosin molecule is composed of 6 polypeptide chains, 2 heavy chains and 4 light chains. The 2 heavy chains, they wrap spirally around each other to form a double helix, which is called the tail of the myosin molecule. One end of each of these chains is folded bilaterally into a globular polypeptide structure called a myosin head. You know why myosin's molecule is called a two-headed molecule? Because there are 2 free heads at one end of the double helix myosin molecule. The 4 light chains, they are also part of the myosin head, 2 to each head. Other parts of the myosin filament, they include body, arms, cross bridges, hinges. A significant property is the ATPase activity of myosin head. This myosin head functions as an ATPase enzyme, and this property allows the head to cleave ATP and use the energy for the contraction process. Let's look now at the composition of the actin or thin filament. The actin filament, they are composed of actin molecules, tropomyosin molecules, and troponin molecules. The actin molecules are composed of F actin molecules and G actin along with its ADP. The backbone of the actin filament is a double-stranded F-actin protein molecule. Each strand of the double F-actin helix, that is composed of polymerized G-actin molecules, and attached to each one of the G-actin molecules is one molecule of ATP, which is called the active site of the actin filament. The cross bridges of the myosin filaments, they attach with these active sites to cause muscle contraction. The tropomyosin molecules are wrapped spirally around the sides of the F-actin helix. In the resting state, the tropomyosin molecules, they lie on top of the active sites of the actin strands. Attached along the sides of the tropomyosin molecules are the protein molecules which are called troponin. Troponin has an important function because this troponin, it attaches the tropomyosin to the actin. Troponin is a complex of three protein subunits: troponin I, troponin T, and troponin C. This troponin I, it has high affinity for actin, while troponin T has affinity for tropomyosin. And finally, troponin C has high affinity for calcium ions to start the muscle contraction. Now, before starting the topic of neuromuscular transmission, I want to give you an overview. We will look at the definition of neuromuscular junction and steps of neuromuscular transmission. You should know what a motor end plate is, then acetylcholine, its formation, transport, and release. These are important topics. You must also know what is end plate potential, and in the end, I am going to tell you about the drugs which affect neuromuscular transmission and a condition which is called myasthenia gravis. The neuromuscular junction is a junction made by nerve ending with the muscle fiber. Each nerve ending makes a junction called the neuromuscular junction with the muscle fiber near its midpoint. You must also know the nerve supply of skeletal muscle. The skeletal muscle fibers are innervated by large myelinated nerve fibers. These originate from large motor neurons in the anterior horns of the spinal cord. Each nerve fiber, after entering the muscle belly, branches and stimulates from 300 to several hundred skeletal muscle fibers. So, what is motor end plate? It is a structural complex formed by synaptic contact of axon with the muscle membrane. And how it is formed? The nerve fiber forms a complex of branching nerve terminals that invaginate into the surface of the muscle fiber. These nerve terminals lie outside the muscle fiber plasma membrane. The entire structure is called the motor end plate. Now, end plate potential or generator potential is the depolarization of skeletal muscle fibers caused by the neurotransmitters binding to the postsynaptic membrane in the neuromuscular junction. It initiates and propagates muscle action potential. End plate potential is produced when acetylcholine gated channels they open due to the arrival of nerve action potential. So there is a sudden sodium influx into the muscle fibers which causes membrane potential to rise in the positive direction. How the end plate potential is affected in myasthenia gravis? Actually, the end plate potential is decreased in myasthenia gravis. The antibodies are formed against postsynaptic acetylcholine receptors. So, acetylcholine is released, but it will not produce an action potential in postsynaptic membrane, or even if it does, the amplitude is low. For understanding neuromuscular transmission, first get to know about the formation and transport of synaptic vesicles and also that what is acetylcholine. Small vesicles are formed by the Golgi apparatus in the cell body of the motor neuron in the spinal cord. These vesicles they are then transported by exoplasm from the cell body to the nerve terminal. Number of synaptic vesicles in nerve terminal of each end plate is around 300,000. The acetylcholine, which is a neurotransmitter, it excites the muscle fiber membrane. In the axon terminal, there are many mitochondria that help in the synthesis of an excitatory transmitter, the acetylcholine. Now, acetylcholine is synthesized in the cytosol of the nerve fiber terminal and stored in synaptic vesicles. About 10,000 molecules of acetylcholine are stored and all time present in each vesicle. Exocytosis is the extrusion after vesicular rupture of acetylcholine into the synaptic space. About 125 vesicles usually rupture with each action potential. The very first step is nerve action potential leading to the calcium ion influx. When an action potential arrives at the nerve terminal, it opens many calcium channels in the membrane of the nerve terminal. As a result, the calcium ion concentration inside the terminal membrane increases. Uh, Next is the fusion of vesicles with end membrane, or you can call it pre-junctional membrane. The action potential causes the synaptic vesicles to fuse with the end bulb membrane, resulting in the release of acetylcholine into the synaptic cleft. So, what is your your second step? That is the release of acetylcholine, in which the vesicles in the synaptic terminal they fuse with the neuronal membrane and they dump their contents into the synaptic cleft. This fusion makes many of the vesicles rupture, allowing the exocytosis of acetylcholine into the synaptic space. And as I already told you that about 125 vesicles they rupture with each action potential. Newly secreted acetylcholine it causes the opening of acetylcholine gated channels. The effect of opening the acetylcholine gated channels is to move sodium ions to the inside of muscle fiber. As here you can see the steps of transmission in the form of a flow chart. The movement of the sodium ions to the inside of the muscle fiber it creates a local positive potential change inside the muscle fiber membrane, which is called the end plate potential. The end plate potential initiates an action potential which spreads along the muscle membrane and causes muscle contraction. Do you wonder that what could be the fate of acetylcholine after performing its function? So far, you know that the acetylcholine excites the muscle fiber membrane. After a few milliseconds, the acetylcholine is split by acetylcholine esterase into acetate ion and choline. The choline is reabsorbed actively into the neural terminal to be reused to form new acetylcholine. Now, the next question is that how new vesicles are formed. You see, within a few seconds after each action potential is over, coated pits appear in the terminal's nerve membrane caused by contractile proteins. The contractile proteins underneath the pit they contract and they cause the pits to break away to the interior of the membrane, thus forming new vesicles. Within another few seconds, acetylcholine is transported to the interior of these vesicles for next cycle. Here I will just give you the names of the drugs that enhance or block the transmission at neuromuscular junction. Drugs that stimulate the muscle fiber by acetylcholine-like action are methacholine, carbachol, and nicotine. Then there are certain drugs that stimulate the neuromuscular junction by inactivating acetylcholine esterase. And the examples include neostigmine, physostigmine, diisopropyl fluorophosphate. The interesting ones are the chloroform drugs that block the transmission at neuromuscular junction. Myasthenia gravis is a serious and sometimes fatal disease in which the skeletal muscles are weak and they tire easily, and there could also be seen muscle paralysis. The occurrence of myasthenia gravis is approximated around 1 in every 20,000 persons. Cause of muscle paralysis in this condition is the inability of neuromuscular junction to transmit enough signals from the nerve fibers to the muscle fibers. Here you can see another scenario which could be your university question, and it is related to myasthenia gravis. You might be asked about the causative factor of myasthenia gravis. So don't forget that it is caused by the formation of circulating antibodies to the muscle acetylcholine receptors. Clinical feature of the disease is muscle fatigue and generalized weakness of skeletal muscles with sustained or repeated activity. And muscles involved are muscles of limbs, extraocular and facial muscles, and the muscles of diaphragm. Acetylcholine receptor antibody test is reliable for diagnosing myasthenia gravis as a positive finding in the serum examination. And the treatment options, they include administration of neostigmine or physostigmine, which allows larger than normal amounts of the acetylcholine to accumulate in the synaptic space. It stops the breakdown of acetylcholine by blocking acetylcholine esterase. Then there could be performed the plasmapheresis, thymectomy, or there could be administration of immunosuppressant drugs, for example, the glucocorticoids.